Okay, so we're moving into the next part of chapter 4. This is a pretty lengthy chapter, so I'm going to try to break it up as much as we can. So we've already talked about prokaryotic cells, and we've talked about similarities between prokaryotic and eukaryotic cells. And now we're going to dive deep into eukaryotic cells. So remember again, eukaryotic cells are going to be cells of organisms that belong to the domain Eukarya, which include animals, plants, protists, and fungi. Those are the 4 kingdoms. And when we look at eukaryotic cells, they're going to have a membrane-bound nucleus in which DNA will be housed or enclosed in. They also have other membrane organelles that we will talk about, like the Golgi and the endoplasmic reticulum and mitochondria and peroxisomes and lysosomes and chloroplasts if we're talking about plants. And these organelles compartmentalize processes that are going to occur in these cells. So for example, cellular respiration, which we'll talk about in a few chapters, that's going to be the process, the metabolic process, in which ATP or energy is going to be generated for the cells of an organism. And that process is going to take place mostly inside of the mitochondria. So that process is compartmentalized. That specific organelle is specialized for energy transformation. Okay, so we'll talk more about that. And eukaryotic cells have several linear double-stranded chromosomes. Compare that to prokaryotic cells that have just one single circular chromosome. So genetically, different. Okay, so pictured here in these figures, we have 2 examples of eukaryotic cells. The top cell is going to be the cell of, for example, an animal. How do I know this? There's a couple of things in here that we'll talk about. Specifically, it lacks a cell wall. Okay, so this is probably a cell from an animal. If you look at our bottom cell here, this is a plant cell. How do I know that? Again, there's a couple of things here. The key thing is the cell wall. What else do I see? Central vacuole. That's going to be a key characteristic of plant cells that we don't find in animal cells. And there's also chloroplasts. So that's going to be another structure only found in plant cells, not animals. So the next thing we're going to do is we're going to take a tour of the cell, and we're going to go through each one of these structures in a little bit more detail. Okay, for all eukaryotic cells, both plant and animals. So let's start here. We'll start at the outside. Again, plant cells are going to have a cell wall beyond this. Animal cells have some extracellular components called the extracellular matrix. But the barrier, right, of every cell, prokaryotic or eukaryotic, is this structure called the plasma membrane. The components of the plasma membrane are going to look different depending on what cell type we're looking at, but in general, this is what a plasma membrane looks like. And this structure is going to separate the inside of the cell from the outside of the cell. So if we look here on this figure, I think you can, let me just grab a pointer. Okay, this region right here, below this structure, this is going to be the cytoplasm, okay, or the internal fluid of the cell. So this represents the inside of the cell, or the side of the membrane that's facing the inside of the cell. And you can see here, pointing right here, it says filaments of the cytoskeleton. We're going to talk about the cytoskeleton in general. But just for now, realize that the cytoskeleton is going to be found throughout the cytoplasm, but it will be found attached to and helping to stabilize, to some extent, the plasma membrane. Okay, kind of interacting with that plasma membrane. So this right here let's use this, from here all the way up to the top here, this is the plasma membrane. And then outside here is the extracellular fluid, or outside of the cell. So let me grab my marker, make sure we're understanding this. Okay, if this were a cell, like the cell we just saw on the previous slide. I'm just going to draw my circle. That represents the plasma membrane, right? The outer boundary. Inside of here would be the cytoplasm. I'm going to put a C for cytoplasm. And outside of here would be the extracellular fluid. Okay. What we've done here in this picture that we've got is we've taken just a segment of the plasma membrane and we've blown it up. Okay, so we're looking at that barrier. And so here on this figure, again, this would be the cytoplasm or the cytoplasmic side of the membrane, and this would be the extracellular fluid, the fluid which bathes that cell. Okay, and again, from here to here, that is my plasma membrane. Or we could say cell membrane. Those two can be really used interchangeably, those two terms. So what is it? It's a phospholipid bilayer. Okay, in chapter 5, which is the next chapter, we are going to specifically focus on this structure, and we'll learn a little bit more about what a phospholipid is and what a bilayer means, but it's pretty much just two layers of these things called phospholipids, and they're going to be oriented in a very specific fashion so that their tails face each other. And we'll talk about why really when we get to chapter 5. Okay? In fact, we already talked a little bit about this in chapter 3 when we were talking about macromolecules. We did lipids. We talked about phospholipids. We talked about how they compose membranes, and we talked about their structure. Okay, but again, I'm going to remind you of all of that a little bit more. Okay, so we've got a phospholipid bilayer. We've got embedded proteins. Again, we're going to talk about the role of those proteins in the next chapter. If it's an animal cell, there will also be molecules of cholesterol, okay, which is a steroid. Again, we'll talk about that in chapter 5. What I do want you to gather from now is the role of the plasma membrane. You know where it's found. It's found surrounding all cells, right? Enclosing the cytoplasm and any other parts and pieces inside of there, right? All of the organelles in the case of eukaryotic cells. And its job is to control the passage of molecules into and out of the cell. It's the gatekeeper. Nothing's coming into the cell or getting out of the cell without first crossing through this barrier. Okay? The plasma membrane we're going to learn is semi-permeable, which means that some things can cross through the phospholipid bilayer on their own without a problem, maybe even quickly. And then some things have a harder time, or some things are completely blocked. They need, if they're going to get through, they're going to need assistance getting through. Okay, so again, I want you to understand the basics of the plasma membrane here in this chapter, and then in chapter 5, we're going to go and take a real dive deep into that membrane. Okay. So remember how we talked about cell size being limited, and that's because we have to have a high surface to volume ratio. There has to be enough membrane to service the inside of the cell. One way that some cells kind of overcome that is by creating these structures called microvilli. So what do? They essentially, so if this is a cell, right, draw a circle. Instead of just having kind of a flat plasma membrane that surrounds the cell, equally, instead what they'll do is they'll take parts of their plasma membrane and they'll fold that plasma membrane in. Almost looks like fingers. What did they do? They just took and they expanded their surface area. They just increased their surface area to volume ratio by a lot. Okay? So there are some cells in our bodies that have to absorb a lot of things or maybe excrete a lot of things. So in this example here, this is a picture from the lining of the small intestines. And on this side, that's an electron micrograph, a transmission electron micrograph of the small intestine. And you can see these structures here. They look like fingers. Those are called microvilli. One is called a microvillus. And here's a cartoon of that. Okay, and you can see that where those microvilli are, that's the inside of the small intestine. And a key function of the small intestine is absorption. And so if we have microvilli there, we've just increased our surface area to volume ratio. And the more membrane there is in that section, the more that will be absorbed by those cells. So here's another example of form equals function, right? Cell shape here is directly correlated to the cell's function. those microvilli are there for the sole purpose of increasing absorption. And that's the job of those cells that line the inside of that small intestine. Okay, so not every plasma membrane is built the same. And again, some cells will alter the shape of that plasma membrane to enhance their function. Okay, so that's the plasma membrane. Again, we're going to come back to that in Chapter 5 and really dive deep. So our next stop on our tour of the cell is the cytoplasm. This is going to be the fluid substance that's going to be found between the plasma membrane and the nucleus. Okay, remember we're talking about eukaryotic cells here, so there's a nucleus. So all that fluid, or sometimes gel-like substance in some regions, is going to be called the cytoplasm. The gel-like substance is called the cytosol. So the consistency of that can change depending on where we're looking in the cell. Organelles are going to be found in the cytoplasm, again, kind of in that gelatinous structure or substance called the cytosol. When we look at the cytoplasm, it's about 70 to 80% water. And that percentage changes depending on what cell type we're looking at. Some cells have a higher amount of water and some a little bit lower, but on average, the cytoplasm is 70 to 80% water. And we find organelles, we find other things like proteins, we find organic and inorganic molecules. The cytoplasm is a site where some reactions take place. Let me give you an example of a reaction that takes place in the cytoplasm. I mentioned cellular respiration already a few times. Again, we're going to talk about that really deep in Chapter 7, but for now, cellular respiration is a process that takes place in the mitochondria of eukaryotic cells, mostly, right? So most of the process happens in the mitochondria. However, the first step of cellular respiration is called glycolysis, and that does not happen in the mitochondria. Instead, this is a series of metabolic reactions that take place actually in the cytoplasm, and they get this process rolling. So the cytoplasm is a really important site for many things, including metabolic reactions. Okay, let's go to the nucleus. So the nucleus, if you're looking through a microscope at a cell, it's going to be the most prominent organelle, easy to stain, real big. Okay, what is it? It's going to be the house of your genetic material. So your chromosomes, your DNA, is going to be housed inside of this double membrane bound structure. Okay, so here it is in this figure. Now in this figure, it's this light purple. All of this is the nucleus here. Here's the outer boundary here. Okay, that dark purple line. What we find outside of here, this blue stuff, from here to here, this is part of what we call the endoplasmic reticulum. We're not there yet. But I do want you to notice that the endoplasmic reticulum is continuous with the nuclear envelope, which is kind of neat. So that membrane that surrounds the nucleus is continuous with the endoplasmic reticulum. We'll come back to that. So this is the nucleus, all the way from here to here, this whole purple region. And all of this stringy substance inside of here, that's all DNA. Okay, now, as a precursor to when we get into mitosis and meiosis, I need you to know that your DNA is never naked. It's never just willy-nilly strung out in the nucleus. Instead, it's always organized by proteins. We'll learn more about those proteins in a little bit, in a couple of chapters. But for now, know that the DNA associated with those proteins is called chromatin. So we call DNA in the nucleus, if it's in a non-dividing cell, a resting cell, we call it chromatin. Okay, so your genetic material, again, is always going to be organized by some proteins, and so it'll be in a form called chromatin. Okay, your nucleus is surrounded by a structure either called the nuclear membrane, or some textbooks will call it the nuclear envelope. It's a double membrane structure, which means that there are two phospholipid bilayers surrounding the nucleus. How does stuff get in and out? It's tough. And it's meant to be tough because you really don't want anything coming in or getting out of your nucleus that doesn't belong in your nucleus or belong being lost from your nucleus. And so anything coming in or out has to move through a specialized structure, which is going to be gated by a protein called a nuclear pore. Okay, and you can see right here, here's a nuclear pore. These are tightly secured pores. It's not just like a hole. They're going to be guarded. So in order to get in or out, proteins will have to recognize what's coming in or what's leaving the nucleus. Tightly regulated process. Your genetic material, that's your blueprint to everything that makes you you, every process, every enzyme that's made. That's all coded for in your DNA. So you really don't want anything getting into the nucleus that could potentially damage your DNA or mess with any processes that are happening inside that nucleus. Now, in the nucleus, there is a fluid, just like the cytoplasm, except for we call it the nucleoplasm. It's a semi-fluid inside that nucleus. And I think the next slide I talk about this, but see this dark spot right here? Let's mention it now. It's called the nucleolus. Okay, yeah. I do talk about it in 2 slides. Let me just give you a heads up from now. The nucleolus is going to be the site of ribosome assembly. Okay, and I'll tell you what those are in a second. I wanted to take one more peek at the DNA. And again, we're going to come back to this when we get to chapter, I think, 10, when we start talking about cell division. But I wanted to introduce you to this a little bit more now. So again, inside of your nucleus, you have your genetic material. And most of you are probably familiar with seeing genetic material like this, in the shape of X's or chromosomes, right? So each one of these is an X. You can kind of see this blue one maybe the best. It's 2 strands joined at the centromere. Here it is in cartoon form. But, by the way, this right here is called a karyotype. This is actual chromosomes that have been stained. It's pretty neat. Here's a cartoon. So right there is a chromosome, right? That's what you're probably used to seeing, an X-shaped structure. We'll talk more about how those form, again, a little bit later, but for now I need you to know that if we were to uncoil or unravel a chromosome, we would be left with chromatin. There's that term. Okay, and chromatin is made up of here. It's made up of DNA, which is the purple stringy stuff, wrapped around or wound around a central core of proteins called histone proteins. Okay, so it's DNA plus proteins. Why? Well, because your genetic material needs to be kept organized. If not, it will become knotted, maybe broken, and that's dangerous, right? We don't want anything happening to our genetic material. So in a resting cell, in a non-dividing cell, your DNA will be in the form of chromatin, and then when your cell goes to divide, it will be in the form of chromosomes. Okay. Let's look at that nucleolus. Remember, that's that dark, densely stained region found in the nucleus. Sometimes there's multiple nucleoli, sometimes just one. In our figure, there was just one. Again, I said that that spot is the site of ribosome assembly. That's where ribosomal RNA, that's ribonucleic acid, okay, and ribosome subunits, those are proteins, where they get assembled into ribosomes, and then they get transported out of the nucleus. Okay, so what's a ribosome? A ribosome is going to be a 2 subunit structure, which is responsible for making proteins. Okay, so the nucleolus is the site of ribosome assembly. It's where they get put together and made. Then they get shipped out into the cytoplasm, and out in the cytoplasm is where ribosomes make proteins. They are essential, really, really important structures. Okay, they're made in the nucleus, inside of that nucleolus. Okay, so here's what a ribosome looks like. So again, the job of the ribosome, you need to know that they are responsible for making or synthesizing proteins. Okay, and what proteins a ribosome makes depends on your DNA. It depends on what genes are being expressed in your cells at that specific time. When we look at a ribosome, the ribosome here is the orange and light blue. It's a 2 subunit structure, named very easily the small ribosomal subunit and the large ribosomal subunit. They're going to come together again in the nucleus. They'll be put together in the nucleus, in the nucleolus. Then they get shipped out to the cytoplasm. And they'll come together, that small and large ribosomal subunit, during protein synthesis. what the ribosome does is it reads messenger RNA, and as it reads the messenger RNA, it's going to start to link together amino acids. And you know that amino acids are the building blocks of proteins, right? So proteins are the functional units in your cells that are made up of amino acids. How do those amino acids get put together? Here it is. This process right here is called translation. And if you take General Biology 2 with me, we will talk in detail about this process. But to sum it up, what happens here is a ribosome reads pretty much a copy of the gene, which is the mRNA. And as it reads the mRNA, based on that sequence, it's going to string together amino acids in a very specific sequence until we end up with a protein. Okay, those amino acids will then fold up in proper three-dimensional conformation, and we end up with a functional protein. So again, you need to know that ribosomes are made in the nucleolus. They do their job in the cytoplasm, and their job is to make proteins. Okay, on to the mitochondria. So the mitochondria, we've already touched on this. I talked about cellular respiration a few times. That's what's happening in the mitochondria. You've probably heard it referred to as the powerhouse or the energy factory. What does that mean? That means that the mitochondria is responsible for making ATP, which is energy. That's the energy currency that your cells are able to use. That process, that metabolic process, is called cellular respiration. In cellular respiration, we're going to take food. Glucose is our prime example of food here. And we're going to break the glucose down, and we're going to extract energy from it, and that energy will be converted to a usable form, ATP. Okay, and again, the bulk of that process is happening inside the mitochondria. I did put the formula for cellular respiration there for you. Glucose plus oxygen yields carbon dioxide, water, and ATP. ATP is energy. Okay, so that's there for you. And again, we're going to get into that in detail when we get to chapter 7. Here's a mitochondria blown up. Mitochondria look kind of like kidney beans. They're pretty neat little organelles. They've got an outer membrane. They've got an inner membrane. And in between them, they have a space called the intermembrane space. That space is really, really important to the process of cellular respiration. The inner membrane is also folded. Okay, and those folds are called cristae. Think about why. Why would it be folded? Well, the more membrane you have, the more surface area you have, the more metabolic processes that can take place. The inner membrane of the mitochondria is a key site for cellular respiration. One of the key processes, called the electron transport chain, is located in that inner membrane. So the more membrane you have, the more proteins involved in the electron transport chain you can have sitting in there, which means the more ATP you can generate. Okay? The inside of the mitochondria is filled with a fluid, and that region is called the matrix. Now you might notice the little purple dots on the inside there. Those are ribosomes. So mitochondria have their own ribosomes. And right here, do you see that little circular structure? That's DNA. So mitochondria have their own genetic information. It's circular, double-stranded DNA, which is pretty cool. You may have heard of the theory of the endosymbiont theory, that mitochondria may have once been free-living bacteria that were engulfed by a cell and became specialized in that function of carrying out cellular respiration. There's lots of evidence. You may have heard of Lynn Margulis. She's the scientist who suggested the endosymbiont theory for both mitochondria as well as chloroplasts. Pretty interesting. Okay, off on a tangent. So here is the electron micrograph of the mitochondria, and again, here's the cartoon. Okay, so again, structure, it's a double membrane, an outer and an inner membrane. Those are both phospholipid bilayers. There's a space in between The inner membrane is folded into cristae to increase surface area. The inside fluid is called the matrix, which houses ribosomes as well as DNA. Okay, I think I covered everything on there. This again is the site of cellular respiration. So we're going to move on next to peroxisomes. So peroxisomes are going to be small, round, membrane-bound compartments or organelles. And a couple of things happen inside of here. This is a site where fatty acids and amino acids will be broken down into their component parts and pieces and recycled by your cells. But peroxisomes are also a site of detoxification. And we're actually going to do a lab where we'll talk about this a little bit more. So when I talk about detoxification for peroxisomes, this is a site where hydrogen peroxide will be broken down into water and oxygen. And we're going to do a lab, again, on this detoxification process. There's a very specific enzyme in the peroxisome which speeds up this reaction. It's a catalyst, and it's called catalase, which is kind of confusing. So catalase, C-A-T-A-L-A-S-E. We're going to do a lab on catalase where we show how it breaks down hydrogen peroxide into water and oxygen, and we're going to look at how different environmental conditions can alter the activity of this enzyme catalase. And catalase is found inside of peroxisomes in eukaryotic cells, and that's its job, to break down hydrogen peroxide. Okay, so we'll be getting to that in a couple of weeks. That's one of the labs where you will be performing a reaction at home. Okay, and one of the ingredients you need for that lab is hydrogen peroxide, which is one of those things that might be a little difficult to find right now. So make sure that you get your hands on that sooner than later. Okay, so after peroxisomes, we're going to talk about vesicles and vacuoles. These are going to be membrane-bound sacs, usually round and small as well. And their job can be a plethora of different things. They might function in storage, so they may store cellular components that can be used later. They might be involved in transport, so bringing stuff in from the membrane, moving stuff between organelles, or carrying stuff out to the membrane. Those would be vesicles or vacuoles that would be responsible for that. Some vesicles may contain enzymes involved in digestion. And then vesicle membranes can fuse with the plasma membrane again, and they can fuse with other membranes in the cell. So if there's a vesicle involved in transport, it will be able to fuse with other membranes inside your cell. I'll show you some pictures of that in a minute. Okay, so I just wanted to point out some key differences between plant and animal cells. Everything that we've talked about so far is shared between plant and animal cells, but there are a few things that are not shared, that these have, there's some differences. So the first one in animals are going to have these structures called centrioles that are really part of the cell's cytoskeleton, which we'll get to in a few minutes. Plant cells do not have those. Okay, animal cells also will have structures called lysosomes, and plant cells do not have those. Okay, what do plant cells have that animal cells lack? Plant cells have a cell wall, they have chloroplasts and other plastids, and they also have this large central vacuole. It's usually filled with water, surrounded by a membrane called the tonoplast. Those things are not found in animal cells. Let's take a peek at some of these. So the first one on the list, this is again a structure that animal cells have that plant cells do not. Animal cells will have this structure called the centrosome, where at the center of it, they will have a pair of these structures called centrioles. And this whole structure is used during cellular reproduction, so during mitosis and meiosis. So this is what the centrioles look like right here. This is the centriole, or the centrioles. There's two of them. These would be at the center of a structure called the centrosome. Okay, when we get to chapter 10 and 11, we're going to look at these even further. So what a centriole looks like is it's going to be composed of structures called microtubules, which are part of the cytoskeleton. And if we look around this ring, you can see that the microtubules are in triplets, and there's 9 of them. Okay. What are those used for? Again, we're going to come back to this when we get to mitosis, but for now, if this was an animal cell here, and it was about to go through mitosis, what there would be are 2 poles, so a pole here on one side of the cell, and a pole here on the other side of the cell. And what I just drew for you, not fantastically, is the centrioles. So here's a set, and here's a set, or a doublet. They would move towards the poles, and then out of them would grow this structure called the mitotic spindle. Now the mitotic spindle is going to be involved in pulling chromosomes apart during mitosis and meiosis. Now if this were a plant cell, this is how the plant cell would look. Well, first off, there would be a cell wall, so I'll draw that. There's also a plasma membrane, but the plasma membrane is usually pushed real tight against the cell wall, so I won't even draw that. Now, there will still be centrosomes at the poles, so there'll be microtubules coming out like this and like this on 2 sides of the cell. But at the heart of the centrosome, there will not be that doublet, those 2 centrioles. They're just not there in plants, and we're really not sure why. Now, we think in animals that the centrioles at the center of the centrosome are responsible for organizing the mitotic spindle, which then again pulls the chromosomes apart during mitosis and meiosis. But the absence of those structures in plant cells doesn't affect the process. It still looks the same. So again, we're really not sure why animal cells evolved those structures and plant cells did not. However, that's one key difference between plant and animal cells. Okay, so the presence of centrioles at the center of the centrosome is only found in animals, not in plants. So again, there's one key difference. Okay, the next thing on our list, let me move my camera here. Animal cells have these structures called lysosomes. I studied lysosomes during my graduate work, so I really like lysosomes. They're one of my favorite compartments. When you think of a lysosome, you can kind of think of a garbage disposal. Its job is to pretty much chew up waste and to recycle it most of the time. Okay, so what kind of waste? Well, that waste might come from inside the cell or from outside the cell. In this particular example, we've got waste from inside the cell. So we're going to come back to this, but when we get to chapter 5. But you can see down here the term, the term autophagy, which literally means the cell eating itself. Autophagy, to eat oneself. So let's take a look at what's happening here. So in purple, we have this structure called the lysosome, and the blue dots inside of there, those represent enzymes. Okay, and then we have this vesicle here carrying a beat up, used, no longer working very well mitochondrion, because there's one, and a peroxisome. So those are 2 organelles that for whatever reason have become they're not very good at working anymore. Okay, and so your cell, it could get rid of them, it could just kind of kick them out, but then that creates waste outside the cell, and that's not always a great idea. So instead, what will happen is the lysosome will fuse with that vesicle. Okay, and so that's happening here, that fusion. And when that happens, those enzymes, those little blue circles in there, they become activated, and they're literally going to kind of chew up whatever contents is inside of there. They're called hydrolytic enzymes. And after they chew up whatever's in there, that digestion, then the cell decides what to do with the chewed up parts and pieces of those organelles. And usually the cell will recycle and reuse those parts and pieces. Okay, up here in this figure, this is an electron micrograph showing this exact process. Okay, so we've actually seen this happening. We've been able to capture it on the microscope. It's pretty cool. Again, we're going to come back to this. So that's what the lysosome is. It's It's a recycling compartment. It contains enzymes that can break stuff down. Okay, so those were some things present in animal cells that we don't find in plants. How about some things we find in plants that we don't find in animal cells? Well, the key structure, the top one, the most obvious, is the cell wall. So plants are going to have, outside of the plasma membrane, they're going to have a rigid structure that can be thin or thick depending on the age of the cell. And what it's there for is to provide structural support and to give the cell a shape. It's made mainly of the polysaccharide cellulose, which we learned about back in chapter 3, and there can be some peptidoglycan as well. Okay. What else do plant cells have? They have central vacuoles. These are going to be large compartments. In fact, many times they occupy most of the cytoplasm of the cell, so they're big. Again, they're surrounded by a plasma membrane, or by a membrane called the tonoplast. It's a phospholipid bilayer. And the job of the central vacuole is to regulate the concentration of water. And again, we're going to talk more about plant cells and the role of the central vacuole when we get to chapter 5. But for now, what I need you to know is that plant cells like to take on a lot of water and be really, really robust. Because of the presence of the cell wall, plant cells will not swell and burst. There is that protection because of the pressure that the cell wall provides. And so the central vacuole helps plant cells to be able to take on the most water that they possibly can. So it's a key structure in maintaining cell shape, rigidity. We have to think about a soft-stemmed plant like like a tulip. Okay, that tulip is standing up straight. There's no bones, right? There's no calcified hard material. We've got cell walls, which are structurally supportive, but really it's water that helps that flower to stand up. If your tulip goes a day or so without water, what happens is it starts to fall over, right? It's the water that helps it to stand up rigid. So that central vacuole is going to be key to allowing plant cells to take on water, which again helps to give structural support to the plant body. Really, really important. Okay. What else? They have chloroplasts. When you think of chloroplasts, I want you to think photosynthesis. So this is going to be the site of photosynthesis in plant cells. I give you the formula for photosynthesis. Again, we're going to come back to photosynthesis when we get to chapter 8, I think. Okay. Photosynthesis is water plus carbon dioxide plus energy from the sun in the form of waves. And that will yield glucose, food, plus oxygen, okay, as a byproduct. Again, it's a double membrane organelle that's compartmentalized. We've got an inner and outer membrane. We've got a central fluid here called the stroma. We've got these disc-shaped structures called granum, or thylakoids, which in a stack are called the granum. This is where photosynthesis will take place. Chloroplasts also, just like mitochondria, have their own ribosomes, and they also have their own small circular DNA, which again leads back to that endosymbiont theory, which is pretty cool. Okay, so that's it for some of our organelles so far, and we've covered the differences between plant and animal cells. So go ahead and get this Are You With Me done, and when you're finished, come back to me. I got some questions for you. Okay, first question. Which of the following is surrounded by 2 phospholipid bilayers? So which one has a double membrane? Ribosomes, vesicles, the cytoplasm, or the nucleoplasm? Okay, so if you need more time, pause me. Ribosomes don't have a membrane. Vesicles and vacuoles are surrounded by a single membrane. The cytoplasm is surrounded by the plasma membrane, which is a phospholipid bilayer, but it's just a single phospholipid bilayer. The nucleus is the only one on here. The nucleoplasm, right? That's the fluid in the nucleus. The nucleus is the only structure on here that has 2. It's a double membrane. organelle. Okay, the only double membrane organelles that we have are the nucleus, mitochondria, and chloroplasts. Those are double membrane compartments. Everything else that has a membrane has a single membrane. Which of the following is both in eukaryotic and prokaryotic cells? Do they both have a nucleus? Do they both have mitochondrion? Do they both have vacuoles? Or do they both have ribosomes? What's present in both eukaryotic as well as prokaryotic cells? Okay, the only one on here that's true is ribosomes. So both prokaryotes and eukaryotes have ribosomes, as well as cytoplasm and the plasma membrane. But prokaryotes don't have a nucleus, prokaryotes don't have mitochondria, and prokaryotes do not produce vesicles or vacuoles. Okay, a couple more. What is the purpose of the folds, the cristae, found in the inner membrane of the mitochondria? Are they there to store food in the form of glucose? Are they there to transport waste products out of the cell? Are they there to make lipids, metabolize lipids? Or are they there to increase the surface area of the membrane, which would then increase ATP production? So hopefully you answered the yellowy orange, right? They're there to increase surface area. Okay, last question, I believe. Nope, there's more. I forgot I put more in here. In the lipid bilayer, blank fatty acid tails face each other within the bilayer and form a core region that excludes water. Ooh, we didn't talk about this. I forgot. So chapter 5, we'll get into these terms, but you know what? This is great. Let's talk about them a little bit now. You're going to see these terms in the next chapter, in chapter 5, and then you'll see them again in lab. So it's really important. So in the lipid bilayer, and we talked about this a little bit in chapter 3, but we didn't really use these terms too much. So remember, you have a head group and some tails, and then that's one layer, and here's the other layer making this a bilayer. Head group and some tails. So I just drew two phospholipids facing each other. Okay, the head groups are going to be facing the outside or the inside of the cell, which is going to be fluid. So they like fluid. So that term, hydrophilic, remember we used it when we talked about water. So hydrophilic, those head groups will be hydrophilic. Now the tails on the inside, the squiggly lines here, they are going to not face the water. In fact, they expel water. And so those fatty acid tails we say are hydrophobic. Okay? So again, in the lipid bilayer, blank fatty acid tails face each other. So the term hydrophobic here in yellow. Okay, we didn't talk about hypertonic or hypotonic yet. We did talk about hydrophilic and hydrophobic back in chapter 3, so hopefully you can recall that. Okay, so those fatty acid tails are hydrophobic. Okay. Upon observing a cell of a grass blade, or a blade of grass, I should say, you notice a prominent, so a large, sac-like structure that occupies a large portion of the cell. Which organelle is this? Is it the Golgi body, the central vacuole, the lysosome, or the chloroplasts? So hopefully you answered blue here. That's going to be called the central vacuole. Last question. Which strategy is most effective for larger cells that have an effective surface area to volume ratio? Should cells have locomotion? Should they move around? Should cells be short and fat? Should they have thin finger-like projections called villi or microvilli? Or should they have thinner plasma membranes? What's a strategy to increase the surface area to volume ratio? So hopefully you answered green there. Right? Anytime you fold the membrane up in microvilli or in cristae, if we're talking about the mitochondria, that's always going to increase the surface area to volume ratio, making that cell more efficient. Okay, I think I'm going to pause here, and I'm going to make a third part for this chapter. So go ahead and start the next video when you're ready.